I've been an archaeologist for 25 years and dug up hundreds of skeletons, worked on loads of excavations, and just found it fascinating that you could learn so much about a person just from the skeleton. On the campus we found 11 skeletons, and they ranged in date from the early Neolithic to the Middle Saxon period. When we analyse skeletons, ideally we need 100% of the skeleton for an accurate age and sex estimation. DNA is able to help us fill in lots of the gaps that, looking at the physical remains, we don't know, we don't understand. For instance, it'll inform us about physical appearance, migration patterns, and pathogens or diseases that don't show up on the skeleton.
I've been studying human evolution for more than 20 years now. So when human remains were discovered on this site here at the Sanger Institute, I was excited. But I had a lot of reservations because you can't get usable DNA from most bones. One of the surprises was that Hinxton is actually a very good site for preserving ancient DNA. So we planned a project involving the archaeologists, and the bone samples were sent to a lab in Australia that's expert in extracting ancient DNA.
As scientists, we often try to make predictions about our past based on modern genomes. However, that signal gets overwritten over time and it becomes a bit more noisy. But ancient DNA gives us this opportunity to go back in time. It's almost like a time machine, and look at those real ancient genomes and see whether our predictions were right or not. In the original study of the skeletons excavated at Sanger, the authors were interested in the pattern of migration and the relatedness of those samples to present-day Europeans. 2 of them were Iron Age males that were shown to be more related to Northern European populations. We can sort of say they were Britons. And 3 of them were Anglo-Saxon females, more closely related to present-day Danish and Dutch populations. We had a look at more physical characteristics. I was able to determine the eye color for most of the individuals. However, in some cases, when the DNA preservation was too low, we were really unable to answer that question.
Studying and sequencing ancient DNA are still quite unusual and quite expensive. That in the future, they should become cheaper and can be applied on a larger scale. And so we'll get a more and more detailed understanding of what happened from a genetic point of view to past populations.
Technology in archaeology is developing all the time. I mean, 100 years ago, it was the introduction of aerial photography, and then there was things like X-rays, carbon dating or C14, and DNA is like that. It's another major breakthrough, another step beyond, which really develops the whole interpretive ability of the archaeologist. To actually be able to look within, to look at the DNA, finding out what their eye colour was, their hair colour was, possibly even where they came from, who their ancestors were, gives you a wider view, a more in-depth understanding of these bones and the people they once were.